This tutorial provides instructions for how to access the biorepository for investigation of neonatal diseases of the lung, or BRINDL. This repository has been created by the Human Tissue Core for the Lung Molecular Atlas Program, or LungMap, funded by the National Heart, Lung, and Blood Institute. The best way to access the BRINDL homepage is through the LungMap homepage at lungmap.net. Click on Request Human Lung Tissue, and this will take you to the homepage of the BRINDL repository. If you are a current LungMap Collaborative member with a BRINDL account, please sign in for your usual access. If you do not have a BRINDL account, please request an account through the photo link that will take you to a brief form that will request such a biorepository account. We ask you to complete this short survey to give us a brief accounting of what your interest and purpose is in requesting a BRINDL account. We ask if you're a member of the LungMap Consortium, your interest and purpose in requesting the account. We ask that you identify a principal investigator, place your own name if that is you, and then provide a biosketch or CV for the principal investigator and/or for yourself as the requester. Finally, provide us with contact information, last name, first name, title and role in the laboratory, email address, and telephone number, so that we may contact you with further information. We also anticipate that most requesters of BRINDL access will be affiliated with an institution and have an institution shipping address that you can complete here. Once you've completed each of those elements, click Submit. You will be presented with this window if you've forgotten any required pieces of the form. Say OK. If you have completed all of the required elements, you will be taken from this survey. If you have not completed the survey and would like to return later, click that button, and you'll be provided with a return code by which you can access the initial parts of the survey that you did complete. With your completion of that survey, this will generate an email to BRINDL that asks for access to a BRINDL account. You should receive an email with instructions and an opportunity to set a password, and then you'll be able to access BRINDL and actually sign in. So let's do that now. Sign in, BRINDL, and your password. This will take you to a page that gives you access to summary statistics for the repository. These summaries are in real time, and so it takes a moment for it to load. This summary page will show you the number of available lung samples by type, as well as the number of available non-lung samples also available through BRINDL. The next element of the summary statistics shows you a flowchart of our whole lung processing plan. And what you'll see here is that in general we take individual lobes of the donor lung. One is formalin inflated and fixed, and then paraffin embedded. Another lobe is paraformaldehyde inflation fixed, and then sucrose cryopreserved, blocked and embedded in OCT and frozen. And another lobe may be inflation fixed with a mixture of just OCT and PBS, 50-50, and frozen and blocked and frozen and stored in that manner. Another lobe is usually biopsied for flash frozen non-fixed blocks. These can be used for protein homogeneous or isolation of DNA or RNA. We also may take fresh tissue from one or more lobes to do micro-CTs. And then at least one lobe has been dissociated into single cells and frozen in a mixed cell dissociation. We also process the trachea and main bronchi in a similar way. Both frozen and paraffin-embedded tissues are available. And then we are open to applying additional preparations that investigators may request. And we can have further discussions about those if you are interested. So you see that the cell vials in this summary graph refers to those mixed dissociated cells, paraffin-embedded, unfixed blocks, OCT blocks, and flash frozen biopsies. The next portion of the summary statistics gives you an idea of the distribution of the donors by sex, age at donation, and age at birth. And then the charts give you more detail in terms of number of donors categorized by either chronological age at donation or gestational age at birth, which are categorized into LGANs, extremely low gestational age newborns, late preterm or term. A chart is available for the neonates, for the infants, and for the children samples, as well as for the relatively few adult cases that have been processed in the same way as the pediatric cases in order to be available as comparison controls. As you see, we also have dissociated cells from spleen and from thymus from some of the donor cases, also from blood. These charts are meant only to summarize the repository. Further de-identified donor information will be provided when samples are selected. If you click on BRINDL at the top, it'll take you back to the home page. If you are still interested in receiving biospecimens from the repository, please click on the image link that takes you to a request form for those biospecimens. This form is somewhat longer and more detailed, by which you can enter a request for tissues. There are instructions here for you to read. We ask for contact information, PI information, a project title, a brief set of specific objectives, and an experimental plan. The experimental plan we ask to be no more than 3 total pages of text, excluding references and figures or tables. Please be sure to read the instructions. The experimental plan description may be in a summary or outline form using bulleted entries, but it should be in enough detail to determine the suitability of the project to use the LungMap resource and the availability and suitability of the LungMap materials for your project. We ask for a summary abstract in non-technical language. We would like to be able to publish this summary abstract on the LungMap website if your application is approved. Your project summary will help us understand your work, facilitate collaborations with you and between you and others who request samples from the repository, and will also inform the public of the important work and discoveries our investigators through LungMap are accomplishing with this resource. We ask for a brief set of specific aims that may be the same or different from the objectives we asked about earlier. We ask for brief background and hypotheses and preliminary data that you think will help us to understand your project and its importance. You may upload a file here to demonstrate that preliminary data if that will be easier for you. Just click Upload and it will give you instructions. We then ask for a summary of your experimental protocols to be applied to the tissues or cells, give you an opportunity to load up to 5, up to 5 references, just the bibliography of those references that may help us to understand your project. We ask for current and or pending funding that will support your proposed studies. These may be pending and they are not required, but they do reassure that the lung map samples will be used. Part H allows you to give a summary of donor demographics to be requested. This is age, if there are specific ages that you know, sex or race, if also these are to be specific or limited. You can also describe specific features of the cases that you will desire and an estimate of the number of donors with this feature. So if you would like all donors to be less than 1 year of age, or this may ask for lung only from cases of anencephaly, which we do have in the biorepository, or other specific features that only you will know. Part J asks for sample types to be requested. Again, if you know these, it will help us to understand if we have available for you what you will need and how many investigators are requesting specific tissue types. So tissue types and their preparation and an estimate of the number of those types and preparations. As you complete these parts of the form, a new section will become available to you to request a second type if that is necessary. We then ask for just a brief justification for tissue type and number requested, and what your expected outcome and scientific significance might be. Again, this form is to make it easy for us to review your application and accept it for distribution of samples to you. So the more information you give us, the better, but we do not expect a full grant proposal here. All right, we also have certain agreements in the use of this precious human tissue that we require. One is with regard to an institutional review board. The first part is a PDF of the study exemption that our Human Tissue Core IRB has provided to us. This is just for your information. These tissues currently in the repository are gathered from generous donations by families who have lost a child, but where the lung can't be placed for transplant. But recovery through the United Network of Organ Sharing, or UNOS, makes it possible to place the organ or tissue donation for research. Tissues may also come from biopsies or explants recovered at the time of organ transplantation. Since the research does not involve intervention or interaction with individuals, and since we take care that any information received related to the case is not individually identifiable and cannot be readily associated back to an individual donor or living person, this research has been designated as research not involving human subjects. And the 45 CFR Part 46 Human Subjects Research Regulations do not apply. But we do strongly suggest that you review your use of these tissues with your IRB or ethics board. In this application, you can tell us where you are in that process. Again, in most cases, this will be considered not human subjects research or will qualify for an exemption. We do suggest that you explore that with your IRB. You do not need to have a current IRB approval in order to ask for tissues. As you see here, this gives you an opportunity to say that your approval is pending, or that your institution has determined that their review is not required. Finally, well, not quite finally, we do ask for a data sharing agreement. This is written here in summary. All data is encouraged to be shared with the scientific community in scientific forums and preferably in inclusion in the lung map breath database, although that is not required. Presentations and publications should acknowledge the LungMap Consortium and biorepository for source material. We ask that you agree to a publication policy agreement. The copyright to data and images on the LungMap site is held by the individual investigators and research centers that contributed them. That would include you if you contribute back the data acquired on the BRINDL biorepository specimens. Please include the LungMap team that generated the data being used in the work resulting from the use of the LungMap datasets. Please also acknowledge that you obtained the dataset from the LungMap and/or biospecimens from the BRINDL repository, along with the date downloaded and a grant number. An example citation is provided for you here. Your acknowledgement will help the LungMap Data Coordinating Center and the NHLBI track and continue to support the community use of this resource and its ongoing collection. We ask that you click that you have read and agree to the terms of the LungMap publication policy. And finally, a material transfer agreement is required prior to our shipping of biospecimens to an investigator. A signed copy will be obtained before the specimens and/or data are distributed. The MTA will be pending with the LungMap. After review and approval of the new project application, the MTA will be routed to the Office of Research and Project Administration at the University of Rochester for execution. The U of R will send you a fully signed copy of the MTA for your record. Please click on the PDF in order to download the MTA with which you can begin to complete the agreement. The last portion asks for lab contact and shipping information. This includes a Federal Express account or something similar by which you can cover the cost of shipping. This is currently the only cost to you for the sharing of BRINDL tissues. We don't have this. If you don't have this information at this point, that is fine. As we get closer to sending you material, we will ask further for shipping information. Once you have completed the form, please submit. If you are not able to complete it on your first go, you can save, and this will give you a return code by which you can return and complete your survey. If you have any questions, please feel free to use the BRINDL email address, which is BRINDL, BRINDL, at urmc.rochester.edu to ask any further questions. Once you submit this survey request, BRINDL and the LungMap Tissue Distribution Committee will review and approve meritorious applications. Approved investigators will then be given access to more details on available biospecimens and be provided assistance to select those that will best meet the needs of the investigator. Again, remember that there is a BRINDL email address, brindle@urmc.rochester.edu. I hope that this gives you enough information that you can get started in requesting tissues from the BRINDL repository. And good luck with your research. Thank you.